Well, good morning. If you can take your seat. My name is Nizar Jarjour. I'm the head of the Division of Allergy, Pulmonary, and Critical Care Medicine. And it gives me great pleasure to welcome you today on behalf of Dr. Page, who couldn't be with us today, and our division to the Department of Medicine Ground Round. This is a very special ground round for us. It is the William Busse, MD, Visiting Professor of Medicine. Many of you know Dr. Busse here, who's been at the University of Wisconsin for his entire career. He earned his bachelor's degree from the University of Wisconsin in 1963, MD in 66, did his residency in 71, and allergy immunology fellowship in 73. Dr. Busse joined the faculty in 1974 and rose through the rank to become a professor in 1984. He served as the allergy section chief for 26 years until 2004, and as our department chair from 2005 to 2009. Dr. Busse basically has chaired every important allergy immunology society in the globe. He, just to name a couple of them, he chaired the American Board of Allergy Immunology in 1995 and the American Academy of Asthma, Allergy, and Immunology in year 2000. He led the development and writing of the asthma treatment guidelines in 2003 through 2007. To honor his many accomplishments, the American Thoracic Society bestowed upon him the ATS Breathing for Life Award, given to one person who provided the most influence in respiratory medicine in 2014. Dr. Busse received the largest NIH award in history, given to the University of Wisconsin to study inner city asthma, most recently in 2014 for $70 million. And he has still additional grant and still active in research. It's really a great honor to have Dr. Busse as one of our faculty. And the department and the division is honoring him today with a named Grand Rounds. It's a very special privilege for me to introduce our Grand Round speaker today, Dr. Monica Kraft, who is a very good friend of ours here, and also an international renowned physician scientist who specializes in translational asthma research. And she is the Department of Medicine chair at the University of Arizona College of Medicine, where she holds the Robert and Irene Flynn Endowed Chair of Medicine. Prior to joining the University of Arizona, Dr. Kraft was a division director of pulmonary allergy and critical care and the director of the Duke Asthma Allergy Airway Center at Duke University in Durham, North Carolina. Dr. Kraft completed her MD degree from UCSF, residency and UCLA Harbor, and was there as a chief resident. Then she went to Denver, where she did her pulmonary and critical care fellowship at the University of Colorado and remained there as a faculty, joining the Carl and Hazel Felt Laboratory for Adult Asthma Research, and became the director of the Pulmonary Physiology Unit at National Jewish from 1995 to 2004, when she was recruited to Duke University to start and lead an allergy asthma research program. There, she continued her research studies on the innate immune dysfunction of asthma and airway remodeling, as well as the evaluation of personalized precision approaches to asthma therapy through clinical trial and basic investigation. Dr. Kraft's work has appeared in journals such as the New England Journal of Medicine, the American Journal of Respiratory and Critical Care Medicine, Journal of Allergy and Clinical Immunology, and the Journal of the American Medical Association, Lancet, and Journal of Immunology. Her work has been funded by the National Institute of Health for over 2 decades. She has served in several study sections and will be at the NIH and will begin her term as National Heart, Lung, and Blood Advisory Council member this June. Dr. Kraft has received the Presidential Early Career Award for Scientist and Engineer by President Bill Clinton in the White House in 2000, and she has served as the president of our premier thoracic society, the American Thoracic Society for Pulmonary and Critical Care Medicine, in the year 2012-2013. She will receive the Distinguished Achievement Award from that society in May of this year. It's truly a privilege to welcome Dr. Monica Kraft to give us Grand Round this morning. And then you have yours.
Wonderful. Thank you so much. I'm very honored to be here as your speaker today, and I'm just make sure this is slideshow format. There we go. Got it. I'm honored to give this lecture. I have known Bill really since I was a fellow, and I want to say, Bill, you've been a mentor to me. You've given me some amazing advice over the years that I remember and taken to heart and tried to live by. And, I just really appreciate all the care you've shown with regards to my career, and you've just always, been a wonderful person to talk to. And we had a lot of fun at the latest Quad AI meeting doing our session together. We had to do a roundtable case presentation, just the two of us, and it was really a lot of fun. So thank you all for having me here today. I've known this group for a very long time, and definitely one of my favorites around the country. So thank you. All right, a few disclosures having to do with research funding as well as consulting and royalties. So today what I hope to talk to you about are some challenges really revolving around guideline-based treatment for severe asthma. So I'm going to be focusing more on the severe asthma space. And with that, I think asthma is a really interesting area right now. I think the understanding of the immunology of the disease has exploded, and I think with that are some very interesting tools that have been developed that we can use to treat our patients. So in my personal opinion, I think it's a very exciting time to be in the field. And really what we're trying to do is use this concept of phenotyping and endotyping to really use precision medicine in our practice where we treat patients. So what is asthma? Well, it's a chronic disease characterized by wheezing, shortness of breath, and cough secondary to reversible airflow obstruction. So what do I mean by that? Well, if a patient uses albuterol, then the lung function can improve. Now, in some patients, it may improve to normal. Some patients may start out normal. Others can't quite get to normal. But there is this aspect of this reversibility that characterizes asthma. But it's not just unique to asthma. We see it in other airway diseases too, but it is particularly prominent in asthma. There's also this characteristic of bronchial hyperresponsiveness. So, for instance, this morning was a little chilly outside. So if any of you in the audience have asthma, go outside, take a deep breath of that cold air. You may experience some chest tightness, some wheezing, cough, and that's that airway twitchiness that is a characteristic of the disease. Or in the springtime, since we're moving into spring. The pollen's out, go out, and if you have allergies, go outside again, inhale some of that pollen, same issue. So that's that characteristic, and really we think that's driven in part by airway inflammation, among other things, and that's been a real focus of research really over the last several decades. So this is just a cartoon in terms of what makes it hard to breathe. And really what happens in the airway is the airway is very abnormal in asthma. There's too much smooth muscle, so there's this tendency to constrict. There's also a lot of mucus, and even this airway epithelium, this layer here, is also abnormal compared to a normal airway. And you can understand how all of these changes can narrow the airway and lead to the symptoms that patients come to see us about, which would be wheezing, chest tightness, cough. So I mentioned a bit about the immunology explosion. I'll talk a bit about pathological phenotypes. And then I want to just take a minute and talk a little bit about some work we've been doing with regard to innate immune dysfunction and how that could impact asthma exacerbations. And then a bit on airway structural changes and remodeling. All right, so these are some examples of some innate immune dysfunction advances in asthma. And I'll show you some data along those lines. So we became interested in surfactant protein and A in asthma. And so most of you think about surfactant with regards to its surface tension reducing properties. Give it to babies who are born in respiratory distress. And that's really the surfactant protein B, C, and the lipid portion of surfactant. The surfactant protein A has host defense properties that are well known, but hasn't really been linked to asthma so much. And part of the reason for that is there's a bit of controversy about the role of SPA in asthma. There have been reports saying that it's decreased in the airway in patients with asthma, and this was a report from some time ago. And our own Dr. Zarjour published back in 1999 that when a segmental allergen challenge is done, so that is where allergen is instilled into the airway through a bronchoscope, that in fact that decreases SPA in the lavage fluid coming from the lung. In 2000, there was a report that SPA is increased in asthma. And of course, we added to the challenge by suggesting that perhaps SPA is dysfunctional in asthma. And this is a publication from our group from 2011. So where does this all land? Well, we had a few publications lately, and one is from the Journal of Allergy and Clinical Immunology suggesting that SPA is decreased, but really in patients who are obese and have asthma. So as the field evolves, it may be that these data are all over the map because we haven't figured out the right patients, really where this dysfunction or these levels change. And so we showed last year that in patients who have obese asthma, and that's demonstrated by this OA, have lower SPA levels compared to those who are obese and don't have asthma. And this is overweight asthma and normal, and lean asthma and normal. And these are levels measured in the airway through bronchoscopy. And this decrease in level correlated with lung function and asthma status. So I won't talk as much about this today, but we think this may have to do with compression of the lung that we see in the obese state, because there's more chest wall mass, the diaphragms may be elevated, and those small airways where part of the surfactant protein is produced in those type 2 cells in the very distal lung get compressed, and in fact we think that compression can actually alter cell function. And so we have a separate project really looking at the ability to look at airway cells, apply pressure in the laboratory, and try to mimic some of these compressive events and look at how it affects cell function. So what do we know? So I think we know the protective role of SPA in acute infectious diseases of the lung is well established. We know that in mice who are deficient in SPA, if they're infected with bacteria like Pseudomonas, they have a very exuberant response compared to wild type. I'm going to show you some data suggesting that SPA mice that are deficient, or SPA deficient mice that are sensitized and challenged with OVA, which is an animal model we use in the lab, demonstrate a very robust asthma phenotype. And then actually there may be some genetic reasons for this. There's a paper from a couple of years ago suggesting that specific polymorphisms of the peptides that compose surfactant protein, there's two peptides, surfactant protein A1 and 2, were associated with infant wheezing and increased risk of asthma. So just a few snippets of data suggesting there may be a connection between SPA and asthma. And so these are some data from Duke University from the late Joe Ray Wright, who was a very important mentor, colleague, and friend to me, and Amy Pasfa. And so these are mice who are deficient in surfactant protein A, who are sensitized and challenged with ovalbumin to create an asthma phenotype. And you'll see that these mice demonstrated much more in the way of eosinophils, which is a cell we associate with allergic asthma, type 2 asthma. You can see there's more in the way of certain cytokines, interleukin 4 and 5, that we think are important in asthma, and higher levels of IgE. So it was really these data that got us intrigued about this connection between SPA and asthma. And so then the data I was referring to earlier suggesting that SPA may be dysfunctional really came from these experiments. So in our laboratory, we can actually culture airway epithelial cells, which I know the group does here as well. And the airway epithelial cells line the lung, and they're really the first responders, if you will, to environmental irritants such as allergens, pollutants, even infections. And so we can culture these cells in the lab and produce an ex vivo model of airway epithelial cell function. And so when we did that and we exposed the cells to mycoplasma, so I want you turn your eye to this half of the slide, to mycoplasma, we can see a very significant increase in interleukin 8, which is a cytokine we think is important in innate immunity. Now, if we take SPA from normal patients, you can actually take it, extract it from the lung through a bronchoscopy with lavage, and you add the normal SPA first and then mycoplasma, you see there's very little IL-8 response. But if you do the same thing with asthmatic SPA, so this is SPA taken from lavage from a patient with asthma or from a group of patients, you can see there it doesn't do much at all. It's very similar to the condition where there's no SPA. So that made us think that, in fact, the SPA in asthma may be dysfunctional. So we summarize this by saying if there's an adequate pool of functional SPA, we associate that with normal lung function. But in the asthma state, we can see low SPA, such as in the setting of obesity, or dysfunctional SPA, and in fact, that's what we see in the asthma state. So the question is, why would SPA be dysfunctional? Well, I mentioned a little bit about genetics, and so we've really looked into this in more detail. So this is a cartoon about the structure of SPA. So it actually looks like a bouquet of tulips. And it's made up of two peptides, SPA1 and 2, and each of these peptides have certain domains. The collagen domain is really important for oligomerization, receptor binding. And we're particularly interested in this lectin domain. This domain binds to carbohydrates, pathogens, and lipids. And in fact, there are key locations in these peptides where certain genetic polymorphisms result in amino acid changes. And for instance, in the collagen domain, it's going to be position 50 for SPA1 and 91 for SPA2. And in the lectin domain, it's position 219 for SPA1 and position 223 for SPA2. And I circle 223 because this has turned out to be rather interesting, in that when we sequenced a cohort of patients for the SPA1 and 2 genes, this 223 location turned out to be very interesting. And what it does is it results in a substitution of a glutamine for a lysine. So one amino acid substitution. So one might think, well, how does that alter the ability of this very large protein where there's trimers and it forms this octadecamer? How can that lead to dysfunction? Well, in fact, we associated this particular polymorphism with some asthma characteristics we think are important. So it turns out for patients who, in fact, have 2 copies of the minor allele, so 2 copies of that SNP that results in 2 lysines, they have lower lung function compared to the heterozygote and the major allele. They have a higher ACQ score, so that means they have more symptoms and lower lung function, and they have more in the way of airway and blood eosinophils. So in fact, this particular SNP is associated with asthma characteristics we think are important. So we were surprised by that, because oftentimes you don't necessarily see one SNP really associate with asthma symptoms and signs like this. So we decided to investigate this in an animal model. So we went back to our SPA-deficient mouse, and we exposed those mice to interleukin-13. And this is a well-validated protocol. Interleukin-13 is a cytokine we think is very important in allergic, or what we call type 2 asthma. And in fact, in those mice who didn't have SPA, they had a lot of airway cells, so a big inflammatory response after IL-13 compared to wild type. And these cells were eosinophils and neutrophils in particular, so it was a mixed response. Now, eosinophils we think about with asthma, but there are types of asthma associated with neutrophils too. And in fact, we also see a lot of mucin. Mucin, this is the mucin gene, is elevated, and this is PAS score suggesting that mucin protein is increased. So when SPA is not around and IL-13 shows up, there's a very exuberant change that goes on. And in fact, other cytokines such as IL-6, which we think is important in what SPA does, and eotaxin, which is important for eosinophil migration, are also increased. And so these are just photomicrographs of the lungs from these mice, and you can see, compared to saline, even when wild-type mice are exposed to IL-13, all this pink staining is mucin. But I think you can see the difference when SPA is not around, what happens when IL-13 comes into the picture. There's lots of mucin and all this pink staining. So a very exuberant response. So it's suggesting that SPA may have a role in really modulating the effects of IL-13. But we like to go back and forth from mouse to man because, we're not mice, and not all things in mice translate to the human condition. So we cultured those airway epithelial cells I was telling you about, exposed them to IL-13, and what we find that's very interesting in the lab is in patients who have asthma, when we take the cells out of the body and culture them, they maintain a phenotype outside of the body in culture, which we found to be fascinating and a really powerful tool to be able to look at some mechanisms. And so we see that if we expose the asthma cells to IL-13, we see more IL-8, and we see more MUC5, which is a mucin gene, compared to normal. And if we expose those cells to SPA first, and then IL-13, we can see there's a decrease in both of these mediators. So we decided we were going to breed some mice that really express certain kinds of SPA. So I mentioned the Q variant, so these are the major allele of that SNP I was telling you about. So it turns out when these mice express 2 copies of the Q variant at this position 223, they're protected against IL-13 exposure. So what you can see here is these are total cells in the airway, and you can see that when you have a knockout mouse, you see lots of cells after IL-13. But these Q mice are very much protected. And the K, which are the minor allele, you can see here, not very protected. So there's some differences in the genetics of SPA and then how IL-13 and potentially other exposures are handled by the lung. So we've gone on and constructed a peptide that's instead of 200 amino acids, which is the length of the SPA2 peptide, it's only 10 amino acids. And it spans that lectin domain that's responsible for pathogen recognition and carbohydrate recognition. And in fact, we are now moving to put it into an inhaled form and develop it as a therapeutic for asthma. So can we replace the bad SPA, if you will, with the good SPA? And so this is some work from a house dust model of mice looking at this airway hyperresponsiveness. So we can look at how responsive the airways are with methacholine. And in fact, you can see with house dust mite, there's an increase in this airways resistance. And when we give the peptide, it decreases significantly. So we found these data very exciting. And then when we go to our human cells, and we expose our human cells to IL-13, whether we use the full-length peptide, a 20 amino acid, or 3 versions of a 10 amino acid peptide, you can appreciate mucin gene expression decreases very nicely. And it's this last one here, this version 3, that we're moving ahead with. So maybe one day I can come back to you and talk about how we can add this peptide either by itself, maybe we can put it in with an inhaled steroid for asthma, or if you think you're getting on an airplane, everybody's coughing and hacking, and you're worried about getting an infection, you might need a little extra SPA as you get on the plane through a spray, who knows. So I think we're very excited about where this could lead us. So more to come. All right, so moving on to some other exciting developments in asthma, I wanted to mention this study by the Severe Asthma Research Program. So there's a lot of focus on genes in asthma as well as other diseases. And so what I liked about this study is this group looked at collections or families of genes. And these are from the airway epithelial cells, from patients who have undergone bronchoscopy, and looked at how these families of genes correlate with clinical characteristics. And so I was intrigued by this because it suggests that it really does take families of genes in many cases to modulate a very complex chronic disease. So this group looked at 4 groups of genes. So these are genes related to mitosis, so cell division and cell growth. And those genes correlated with inhaled steroid use and oral steroid use, negatively correlated with quality of life and lung function. So the higher the expression of these mitosis genes, the lower the lung function and quality of life, and the more steroids patients would use. The T2 genes are the ones we talked about having relating to interleukin 4 or 5, some of those cytokines we see with allergic asthma, and we can see positive correlations with race, BMI, inhaled steroid use, negative correlation with asthma quality of life. They also looked at a group of genes responsible for remodeling and repair of the airway, and found that the more of those genes were around, the higher the quality of life and lung function. So those were a good family of genes. And then this is an interesting group of genes really talking about neural function, which is something we don't think about as much in asthma, and saw some positive correlations with quality of life and lung function when those genes were expressed in high levels. So I was intrigued by this study suggesting that these families of genes could really modulate or associate with important clinical characteristics. All right, so another area I want to talk to you about just briefly is CLARA, or club cell secretory protein. So this is a protein that's made by non-ciliated airway epithelial cells, so made by the lung. It is thought to be protective against exposures, although the precise mechanism isn't known yet. We can measure it in the blood. We know that levels are low in smokers. And in fact, there are some associations with asthma and COPD I'd like to tell you about. So in Tucson, we have this wonderful longitudinal cohort called the Children's Respiratory Study. And we also have an adult cohort as well that's been followed for about 20 years. The Children's Respiratory Study started in the 70s and really has been following kids. Really, now they're not kids anymore. And so these children would come at regular intervals for visits and have questionnaires, lung function, blood drawn. So it's an amazing biobank where one can go back and ask questions about early life events and how that may impact the adult situation, especially in folks who have airway disease. And so Fernando Martinez, who directs the Asthma and Airway Disease Center in Tucson, and Stefano Guerra, looked at serum levels of the CC16 protein in kids who were 6 years old, and showed that low levels at age 6 correlated with low lung function later in life. And that low lung function could either be asthma, depending on whether there were allergies involved or certain infections, or COPD if the patient smoked. So really important biomarker. And this has been identified in other studies as well, but not to this degree. And so we've just put forth these data. We're going to present them at the American Thoracic Society meeting this year. We decided to mine a database of messenger RNA or genes, gene expression, from an airway epithelial cell cohort in the Severe Asthma Research Program, what I was showing you earlier, that same database. And we found some amazing associations with the gene that controls CC16 and important asthma outcomes. So we found that the gene that controls CC16 levels, when expressed in the epithelial cells, correlated with asthma status as well as severe asthma. We found associations with lung function, so that means the lower the gene expression, the lower the lung function in asthma. And I think what we were particularly intrigued by is the lower the expression, the more emergency department visits patients would have and the more oral steroids they needed. And I have to say, I've not seen a SNP be this powerful with regards to these kinds of outcomes. So we're really excited about these data, and really we're looking at it as a really important biomarker in asthma. And so we're developing that with others. So more to come in that arena. And then we actually found a very interesting association between low levels and exposure to Mycoplasma pneumoniae, which is the bug that causes walking pneumonia. It's also an important bug that can exacerbate asthma, that can cause asthma attacks. We found that if a person had been exposed to Mycoplasma and had antibodies and had low levels of CC16, they were 8 times more likely to develop asthma. So we found that to be a very intriguing association. Now we're looking at some animal models to understand what CC16 does in the airway. And we have a knockout model for CC16, and we're looking at some very interesting remodeling mechanisms there. So I'm going to switch gears and talk more generally, what is asthma? So it's really a syndrome that's composed of symptoms, lung function, asthma attacks potentially, early onset, late onset. There's different kinds of inflammation, and at the end of the day, it ends up with these are different kinds of asthma or phenotypes. And so really the idea is we want to use precision medicine and really personalize our treatment of asthma. And we're doing this in a lot of other diseases, so asthma is not unique in this way, but I think we're moving ahead, and we're at a particularly nice place with our ability to do this. So I want to talk to you a little bit about how we do it and how potentially you can translate some of these to the clinic today. So when I say phenotype, what do I mean by that? That is a collection of clinical characteristics, basically, that defines a patient. It could be lung function, it could be symptoms, it could be age of onset, gender, BMI, a collection. And they're based on, they don't always necessarily suggest a mechanism, because there could be more than one mechanism driving this phenotype. And really, at the end of the day, we want to get to this endotype. So this is a certain biological pathway that's really driving that phenotype, because if we can figure that out, then the idea is we can develop or choose a therapy that targets that pathway and really make a difference in that patient's asthma. So that's really the goal. And so where are we today with asthma phenotypes? So what you can see is right now we put them into 2 buckets. We talk about the T2 pathway, the type 2 asthma. So that's what I was talking to you about earlier. Typical allergic symptoms are very common, associated with cytokines like interleukin 4, 5, and 13. And then there's the non-type 2. And I'll talk a bit about that in a minute. So under this type 2 bucket, we see allergic asthma, which is probably the asthma that many of us are most familiar with. So it starts early in life, associated with allergy. It can become severe, or it can stay mild. It can go a lot of different directions. There's exercise-induced asthma. There's aspirin-associated respiratory disease, which can actually associate with nasal polyps and frequent asthma exacerbations. And then there's this very late onset type of asthma, usually in women in their 50s, who have eosinophils, and it can be a very labile, difficult-to-treat asthma, often associated with nasal polyps as well. And then we move over to this non-type 2 asthma. So this is not so much associated with the eosinophil. And this, in my opinion, represents a real unmet need in the field. And under this bucket, we see obesity. Now, obesity is interesting, and that obesity can make allergic asthma worse, but it can produce its own inflammatory phenotype, often associated with interleukin 6 and metabolic syndrome and diabetes. And then we have these other types of non-type 2 asthma. We know about 20% of our patients with asthma smoke in the U.S., and so that's associated with a certain kind of asthma, often manifested by neutrophils. There's some asthma that doesn't have a lot of inflammation, that really has a lot of extra smooth muscle and a tendency to bronchoconstrict. And then there's asthma associated with exposure to pollutants and infections. And those aren't necessarily always eosinophil associated. So you can see that there's a lot of different flavors of asthma, and so I feel like our charge as clinicians is to figure out what kind of asthma does this patient have and how can we best treat him or her. And so this slide just reminds me to tell you that of those phenotypes I described, there's a lot of different mechanisms driving them. So I'll give you an example. So if we look at this exacerbation-prone asthma, so that's a kind of asthma that's associated with a lot of asthma attacks. Well, you could have lots of reasons for that. You could have allergic asthma driving it. You could have aspirin-sensitive asthma. You can have late onset eosinophilic. You can have viral exacerbated asthma or even premenstrual asthma. So all of these kinds of asthma can be producing this phenotype. So that's the challenge I think that we're faced now in 2018 is to figure out what kind of asthma are we dealing with, what pathway is driving it. And so these are the slides that can put you to sleep at this time of day, so I will not go through them in great detail, but just to show you really the complexity of the situation. So these are mechanisms of this eosinophilic asthma, and you can see allergens are a big part of that, irritants, even infections, and leukotrienes. That's a mediator that's associated with aspirin, exacerbated respiratory disease in particular. And I think you can see that each of these leads to their own pathways, of which there's some overlap. You can see IL-4, 5, and 13 being important, IL-17 being part of this. So there's lots of different cytokines involved. And then for the non-eosinophilic, some of the triggers are similar, but they result in different pathways. And so you can see that microbes are a very important part of this, oxidative stress, and you can appreciate also infection, air pollution can lead to a lot of different pathways and a different set of cytokines. So what can we do about this in the clinical arena? How can we identify a certain endotype of asthma? So we have some tools available to us and some that we're developing. So sputum eosinophils are nice. It's a great reflection of the airway, inflammation, but they're not the easiest. It's not the easiest test to do in the clinical arena. So we do it as part of our research lab, but it's not necessarily clinically available. So what do we have instead? Well, we have exhaled nitric oxide. That's something we can do to give us a sense of untreated type 2 or allergic inflammation. We can look at blood eosinophils, and we do that. We can look at IgE, the antibody we associate with allergy. We can do allergen skin testing, and we have to collaborate with our friendly allergist to do that. Periostin DPP4 are two biomarkers that are associated with IL-13 expression in the lung. And so those are still under development. They're not necessarily ready for prime time. CC16 I mentioned. And then this other one here, eosinophil peroxidase, which is very interesting. It is a product of eosinophils associated with activated eosinophils. So that would seem like something we'd want to know about. And in fact, you can measure eosinophil peroxidase, nasal swab or throat swab. And there's a group at Mayo Clinic Scottsdale that I'm collaborating with that's moving to develop this as a point-of-care biomarker for the clinic. So wouldn't that be great to know when your patient's right in front of you whether they have a lot of eosinophils or not? Because that you often are charged with knowing that in terms of figuring out, well, are they going to respond to steroids or not? And so it's a very helpful entity to know. So they're almost ready to put it into the clinic, and we're part of a severe asthma clinical trials network called PRECISE that I hope we can bring this in and try to validate it. And these are just some data from that group suggesting that this is nasal EPX and pharyngeal EPX correlates very nicely with induced sputum eosinophils. And so this is a small study, so clearly larger ones need to be performed to really validate the use of this, but I think this holds a lot of promise. So, just to tell you a little bit about what treatments are approved or under development. And this is for the type 2 asthma. This is for the more allergic asthma that we're more familiar with. It's a very crowded space. In fact, the blue boxes are those that are under development. The red are what's approved. So you might have heard of omalizumab, which is an anti-IgE monoclonal antibody that binds to IgE and takes it out of circulation for severe asthma. That's been around for over a decade. We also have inhibitors of interleukin-5. We actually have 3 of them approved right now, if you can believe that. So that's gotten very crowded. And we will have a biologic that really hits both IL-4 and IL-13 in the very near future, and I think that's going to be incredibly helpful. But I think you can appreciate through all of this that there's a lot of action going on in this T2 or this type 2 asthma space. But if we look at the non-type 2, you can see it's a little less crowded, not as well developed. And really we don't have, other than bronchial thermoplasty down here, which is a really non-biologic approach to treating asthma, we don't have a lot of things approved. There's certainly some things under discussion, and as part of our severe asthma network that I mentioned to you, PRECISE, we're going to be looking at things like Gleevec or imatinib, anti-IL-6, and trying to repurpose a lot of some of the biologic use in the rheumatoid arthritis or in the autoimmune space to severe asthma, because there are some similarities in the inflammatory response, especially in the non-type 2 asthma space. So I want to end with a new concept. I'm talking to you about all the ways we can identify what kind of asthma our patient has, maybe what treatments we might select, but can we back it up and do anything to prevent asthma? Wouldn't that be the ultimate? I guess it would mean I would have to find something else to do, but that's okay. So there's some intriguing work going on nationally that is led by my good friend and colleague Fernando Martinez at the University of Arizona that I want to tell you about. So there's a bacterial extract. It's called bronchovoxome. It's approved in Europe. We're going to call it OM85BV for our purposes. It's a series of bacterial extracts, and you can see it's H. flu, Klebsiella, staph, strep, Neisseria, and it's a dried extract which is a mixture of acidic proteins, peptides, and amino acids. And so this has been approved in Europe for decades, really to prevent colds. And in fact, it's also been used to prevent acute symptoms of cystic fibrosis and COPD. And it's been very well tolerated and used for a very long time. So what's the relation of this to asthma? Well, there's a lot of interest, as you might know, in the microbiome in a lot of diseases. Asthma is no exception. So can you somehow use this extract to alter the GI microbiome, and would that have impact on the lung immune system? Because we know there's actually a very tight relationship between those two. And so what do we know about it? Well, there was a small study out of Turkey suggesting that use of this bacterial extract preparation reduced wheezing episodes in children. So that was interesting. The adverse effects were quite low. There was maybe a little bit of GI upset, but in general very well tolerated by both children and adults. And this has also been looked at in animal models to decide, well, how is it really working? How is it changing the microbiome? And then how is it changing the immune system? And it turns out in an animal model, it looks like it's increasing a cell that we think is very important to put the brakes on inflammation called the T regulatory cell. And so, in fact, this bacterial extract increased Tregs in this animal model of asthma. So maybe that's one of the ways that it works. So, led by the University of Arizona, this is a national, multi-center, NIH-funded trial that the University of Wisconsin is participating in, with Rob Lemansky as the PI here, looking at over 1,000 children aged 6 to 18 months who are at high risk for asthma. So these are folks who these are children whose parents might have asthma, they may have atopic dermatitis, they may have wheezed with infection, so they have a predisposition to asthma. So they're going to be receiving this extract for 2 years, 10 days a month, for 2 years, and then they'll be followed for an additional year. And the question will be whether or not this approach can actually delay the presentation or eliminate it entirely of asthma. So this is very exciting, and that, again, a way of really looking at altering the microbiome at a young age that can have effects on the immune system in the lung and alter the presentation of a chronic disease. So we're very excited about this. I think that the trial is now underway. So we're very hopeful that the group can do a good job recruiting these kids and see really if this hypothesis bears fruit. So I think hopefully you agree with me that our approach to the biology of asthma heterogeneity has improved dramatically. I think there's a recognition that asthma is very heterogeneous. It's not just one disease. There are different flavors of asthma, and that the use of clinical characteristics, biomarkers, and responses to treatment will really further hone our ability to deliver personalized therapy and practice precision medicine, which at the end of the day, we want to give our patients the right therapy at the right time. And I think that the question is we honestly need probably more available point-of-care biomarkers to make some real-time decisions regarding therapies for our patients. And as you can see, there are several under development. So maybe I'll be able to come back to you in a few years and give you an update. So thank you for your attention.
Thank you very much, Dr. Kraft, for a wonderful, wonderful grand route. I'm going to ask you to raise your hand, and Dr. Kraft will call on you. Ask your question. I'm going to ask her to repeat the question for the recording.
Dr. Busse. Exactly. This quandary, yes. So the question is really about good bacteria versus bad bacteria. So before I came to see you today, we've done a lot of work with bacteria such as Mycoplasma pneumoniae that aren't good for the lung. And in fact, we showed that there was more of it in patients with asthma, in some patients with asthma. And then, in fact, if those patients were treated with a macrolide, and either clarithromycin or azithromycin, their asthma got better. So that's one kind of situation with bacteria. But now I'm standing here saying that maybe some bacteria aren't so bad. And it turns out that's exactly right. They're not necessarily all bad or all good, but there are groups of bacteria that are actually protective. And of course, lactobacillus being one group, proteobacteria being another. But the lactobacillus are particularly interesting in that the probiotic approach to asthma hasn't really borne fruit, and that there have been studies of probiotic use in asthma that haven't really shown much. But it may be that there are particular species of lactobacillus, and there's one called Lactobacillus johnsoni that has been shown in animal models to really protect against infections and been particularly efficacious. And so the question really is, are there going to be certain groups of organisms that are going to be protective? And in fact, the bronchovoxome, the OM85BV that I showed you, one of the outcomes of treatment is to look at the stool lactobacillus content and look at all the different species, including Johnsoni, to look at its effect. And that may be one way it changes the microbiome. So I think there's sort of good bacteria and bad. And clearly there are going to be viruses and bacteria that exacerbate asthma. But the question is, if we can populate the GI tract with the good bacteria and change the immune system of the lung away from allergic disease, that's really the goal of this approach.
Other questions?
Yes. Hey, Lynn. That's the $6 million question. I know we've been fascinated by that because we were surprised that a 10 amino acid peptide would have the same function or same ability to attenuate inflammation both in an allergen model as well as an infection model, the same as a octadecamer, which is multiple 200 amino acid peptides. So we think because of that lectin domain, which is a critical area of pathogen and carbohydrate receptor recognition, is why. But we're actually doing some competitive binding studies right now to see how it competes with native SPA as well as other agents to really bind to the carbohydrate receptor or to the pathogen. One of the reasons may be also because it's a 10 amino acid, you can deliver a lot more to the lung as opposed to the full-length peptide. And so, in fact, you're getting a lot more of that really active area. And so that may be a simple numbers game, or there may be something about the glutamine-lysine change that actually can alter its ability to compete with native SPA. So more to come on that. We're actually investigating that. Yes. Hey, Jim. Thank you. Yes, thank you. So the question is whether looking at biomarkers such as the CC16 protein in blood and how it correlates with lung expression, and thinking about it, how are we going to use it in asthma? And so yes, there is actually a correlation. We're looking at that in a little more detail now. We have a grant with Stefano Guerra where we can look at serum levels as well as look at airway epithelial cell expression directly. And we're looking at nasal epithelial cells because they're a little bit easier to access compared to lung epithelial cells. There seems to be a relationship, though, between the two. And so I think we can, and that's the beauty, is that we can use serum levels potentially to really provide an assessment of risk for asthma at a young age would be one way this could be used. You can also increase CC16 levels by using vitamin D. Vitamin D increases levels. So that's intriguing to us, and we're thinking about a trial where we actually do that and try to increase levels in those patients who are low and how that may impact lung function. And that would be another way. And then recombinant CC16 exists. It's made by a company in New Mexico. And in fact, there have been some studies in allergic rhinitis that haven't been all that compelling, but we think maybe that's not the right disease process to look at, and the way that it was provided and the patients studied maybe weren't quite the right group. But that's something we're also investigating, is whether we can replace CC16 in those patients who are low and look at how it impacts disease.
So, Monica, I have a question. You mentioned our colleague in dermatology using monoclonal antibodies and other immune-based therapies. And in asthma, we seem to be using them way too late, or only in that severe, extreme kind of disease, while our rheumatology colleagues have now started using it well before there is permanent damage. Do you see that at one point we're going to try to learn from them?
That's a very interesting thought. So, these biologics target specific pathways, so that's good. They're not a steroid. For those of us, especially who take care of patients with severe asthma, we have a love-hate relationship with steroids. They do a great job in the acute setting, but long-term, they have side effects and they're a real problem. And I don't think that's entirely appreciated by the medical community, how much of a negative impact steroids can make in this group of patients. And remember, asthma affects 10% of the population, and in fact, about maybe 10 to 20% of that group, depending on how you look, may have severe asthma. It could be less depending on the setting. And so the question is, can you intervene earlier? I love the concept of that, because if you can really stop some of these processes even earlier in life. I know there's a study going on of which University of Wisconsin is involved to give an anti-IgE in 2 to 4-year-olds to see if, in fact, does that change the whole ability to respond to allergen, improve immune tolerance, all those kinds of questions. And so that's exciting to see if we could intervene early. I think the big issue is going to be cost, because these biologics are not cheap. And so can you justify, from a pharmacoeconomic perspective, using them earlier in life? Now, if you look at hospitalizations and the healthcare costs that patients with severe asthma require or use, they're huge. And so I think there could be a pharmacoeconomic approach to really deciding if it is a good idea to start intervening earlier. So I'm in favor, but I want to be cognizant of cost associated with doing that, and would want to do it thoughtfully.
Question. I have one more question for you. You mentioned chlamydia, and you were among the pioneers in looking at the effect of infection in asthma, and you mentioned some of the downstream impact on SPA. Where are you now with regard to the effect of infection, chronic infection, on the chronicity or severity of asthma?
Right. So, it's funny that you asked that, because it sort of took a move to Arizona to kind of put this all together. So years ago, we looked at mycoplasma and we looked at chlamydia, or chlamydophila, or whatever it's called now, and found that it was in our cohort that there was an increased expression or presence of these organisms by PCR. Not everyone found that around the country, so there was controversy. So why would we see it in some parts of the country and not others, or what kind of patients were we looking at versus other groups? And I wonder now if it has to do with the CC16 levels. And in fact, had we known what we know now, to go back and measure CC16 levels, because clearly there was that interaction with mycoplasma and low levels. And in fact, that may be really the key issue that we didn't know about back in the 90s when we studied this. So it may be a two-hit situation where you need to have low levels of this protective protein and then be exposed to certain infectious agents, and that leads to the presentation of the disease. So more to come on that. We're excited about that possibility.
Any other question? If not, thank you very, very much, Dr. Kraft.
Thank you.
And I have one more task. It gives me great pleasure to present you with the William Busse Visiting Professor of Medicine, a plaque from the University of Wisconsin-Madison, March 30th, 2018.
Wonderful. Thank you. Thanks very much.
And happy Doctor's Day.
Oh, that's right. Yes, happy Doctor's Day. Thank you,
Thank you.
everyone. Thank you.